Hello and welcome to another Mr. Carter Science Special. In this video we'll be looking at how drugs have been discovered from traditional drugs right through to modern sources of antibiotics and other medicines that we can use to treat diseases. So traditionally drugs were extracted from plants or fungi and pressed directly onto a wound that you may have been suffering with. One example of this is the medicine traditionally derived from the plant the willow tree, and we've got a willow tree here. So the bark of the willow tree would have been given to people to chew or to place on their foreheads if they were suffering from a headache. Now it turns out that the active ingredient in the bark of the willow tree is called acetic acid, and that's the chemical that we can now synthesize and package and prescribe to people with headaches as the drug aspirin. So aspirin is a particularly useful medicine if somebody has a headache, but the original compound is derived from the willow tree, acetic acid. Another example of medicines that are derived from plants is the medicine Digitoxin, which comes from the foxglove plant. Now in large doses, Digitoxin is a poison that will stop your heart beating. But in small doses, Digitoxin can be used to treat heart disease by slowing the beat of the heart. And in fact, here is the chemical structure of Digitoxin and the foxglove plant that it's derived from. Now you may have heard of antibiotics, and in fact I have mentioned them in my another video about antibiotics and painkillers. I'll put the link for that below. So antibiotics were discovered by this gentleman here, Alexander Fleming, and Alexander Fleming was working on a plate where he was trying to grow bacteria, but he ended up with a contamination of fungus that you can see here that I've just coloured in. He was very interested to see that there seemed to be a zone surrounding the fungus where there was no growth of his bacteria species that you can see below here. He was very curious as to why that might well be, and he began investigating to see whether or not the fungus produced a particular chemical that seemed to inhibit bacterial growth. In fact, he came up with this particularly arrogant but very accurate quote. "When I woke up just after dawn on September the 28th, 1928, I certainly didn't plan to revolutionize all medicine by discovering the world's first antibiotic or bacteria killer, but I suppose that's exactly what I did." And he certainly did revolutionize all medicine. Now we do not fear bacterial disease as we once did in the past. So penicillin was the world's first antibiotic, and it's named after the fungus penicillium. So Alexander Fleming observed that the fungus penicillium seemed to produce a chemical that prevented bacteria from growing, but it was two other scientists, Ernest Chain and Howard Florey, who found a way of producing large enough quantities of the chemical to treat illness in one particular patient. The patient had a disease known as septicemia, an infection of his blood, and he was hours from death. However, upon receiving an injection of the penicillin antibiotic, the patient began to recover from his blood infection, but he later died when the supply of the antibiotic chemical ran out. But the chemical was so successful in that first trial that scientists went on to try and work out other ways of producing large enough quantities of this antibiotic, and it would go on to treat millions of diseases in patients around the world. So what does the future hold for medicine? Well, overuse of antibiotics means that bacteria species are increasingly evolving antibiotic resistance. So there's a continuing drive to find new medicines that can treat bacterial diseases. Many drugs are now synthesized by research chemists working with chemical banks and computer models. But other potential sources of medicines may be plants or microbes. Potential compounds could be modified to increase their effectiveness. And rainforests or coral reefs are potential sources of plants and microbes that have yet to be tested for medical use. So conservation of these unique habitats is incredibly important in order to utilize the organisms that live there to see if they have any potential medical uses in the future. One example of this comes from Costa Rica, and that is known as the noni fruit. And we can see some examples of the noni fruit here. The noni fruit is used in traditional medicine in Costa Rica to treat communicable and non-communicable diseases. Research has shown that the fruit has some antibiotic properties. However, more testing will prove if the noni fruit can be a viable source of medicines for the future. So there are a number of chemicals from the noni fruit going through drug trials right now. And again, I will put in the links below a link to my video on drug trials. Soil samples may also be a source of new microbes that perhaps could produce antibiotics, but this is very difficult because only 1% of microbes from the soil have been cultured in the lab. They seem to want to have very specific conditions that are difficult to replicate on an agar plate. However, in 2015, a new antibiotic was discovered from a bacteria grown in a very carefully controlled soil sample, and the early indications are that this antibiotic may destroy all bacterial species. Well, that could be very useful for treating bacterial diseases in the future. I hope you've enjoyed this video on discovering drugs and where we identify and find our drugs from. If you have, don't forget to like and subscribe and comment below to let me know what you have learnt. Thank you very much for watching.